Do you ever feel like this some days? Just strung out, stressed, and worried? Well, you're definitely not alone. 87% of adults agree that the past 2 years have been marked by one crisis after another, which all cause significant stress. And when experienced for prolonged periods, this takes a toll on your body. Prolonged stress has been linked to multitudes of illnesses and diseases, like depression, cancer, and stomach ulcers. This is because stress impacts various systems of the body and mind, though the specifics are still a little fuzzy. Knowing this, it then begs the question, what bodily mechanisms are being disrupted to allow for the occurrence of disease? Hi folks, my name is Cole, and I have a Master's of Immunology. Today on Investigate, Explore, Discover, we're going to be looking at how stress negatively impacts immune mechanisms. So hang around with me throughout this whole video to get all of the relevant background information so we can dive into some exciting experimental results. Plus, there's more information for you in the description below. Now, stress is ubiquitous and arises from multitudes of situations. Just like there are many stressors, there's no definition of stress that everyone accepts. Commonly, it's defined as any situation that gives us distress. However, stress is not always harmful. In fact, increased stress can result in greater productivity up to a point. Acute stress is a type of stress that causes a fight or flight response, and it lasts a few minutes or hours. It can be experienced as eustress, like when you're preparing for a date with your significant other and when you get into the zone for work. Or it might be experienced as distress, like what occurs when you're startled by a prankster or a tight deadline. Either way, your bodily responses are controlled by your nervous system, which is made up of different parts. Now, the central nervous system includes the brain and spinal cord, while the peripheral nervous system is split into the somatic and autonomic nervous systems. The autonomic nervous system, which as the name suggests is involved in a number of typically automatic regulatory functions, is then further split up into the sympathetic and parasympathetic nervous systems. The sympathetic nervous system is derived from the spine and controls diverse biological processes, redirecting blood towards the heart and skeletal muscles while providing oxygen and nutrients for a fight or flight response. Most tissues are innervated by sympathetic nervous system fibers, and many cell types respond to neurotransmitters. The neurotransmitters secreted by the sympathetic nervous system are called catecholamines. This group of chemicals includes dopamine, epinephrine, and norepinephrine, also called adrenaline and noradrenaline. Notably, noradrenaline is used as a vasopressor, which increases blood pressure, for treatment of patients with cardiogenic or vasodilatory shock, which includes septic shock, and is normally administered for prolonged periods until the shock subsides. This functions because noradrenaline is recognized by cell surface G protein coupled alpha or beta adrenergic receptors. When activated, they regulate the contractility of smooth muscles on blood vessels to modify blood pressure, among other responses. Now, these receptors are present in many tissues in the body, like the skin, lymph nodes, and the liver. In these organs, the sensory nerve fibers intertwine around the blood vessels and infiltrate into the lymphatic vessels and regions. This is important to note because the lymph node is a sort of central hub for immune cell activation, where antigen is presented to other immune cells to get trained, meaning that there is a link between your nervous and immune systems. Leukocytes is another term for our immune cells, and they traffic continuously around the body via the blood and lymphatic systems. The locomotion of these cells is essential for a functioning immune response and is affected by the physical microenvironment as well as multiple signals which encompass adhesion and chemotactic factors, metabolites, and tissue oxygenation, which the cells use effectively as road signs to figure out where they need to go in response to injury or infection. Now, our immune system is made up of numerous cells that all have particular functions. It is primarily grouped based on the type of activity that the cells have. Innate immune cells, such as neutrophils, macrophages, and natural killer cells, are quick to respond to pathogens and injury and have a non-specialized response. This response is bridged by antigen-presenting cells like macrophages and dendritic cells to prime our adaptive immune response. Our adaptive response is made up of antibody-producing B cells and our cellular-responding T cells, which are critical for our immunity. Now, there are two major subsets of T cells: our cytotoxic CD8 T cells, which kill infected or cancerous cells, and our helper CD4 T cells, which help prime CD8 responses and prime B cells to produce antibodies. Furthermore, when T cells move about, they typically move in an amoeboid fashion, extending pseudopods by rearranging the actin and myosin molecules inside of the cell to take it where it is being directed by the extracellular road signs. Of note, these responses can be influenced by calcium signals. Now, we already know that stress-induced activation of the sympathetic nervous system can influence immune responses. Acute adrenal receptor stimulation increases neutrophils and natural killer cells in the blood, possibly preparing them for injury or infection. It also causes decreased macrophage-induced inflammation. Conversely, sympathectomy of mice increases the CD8 T cell response during influenza infection. Now, this does not even mention the increase in reactive oxygen species that occurs in people with chronic stress. Ultimately, these diverse observations underlie why a mechanistic understanding of cellular function is needed when assessing the impact of sympathetic nervous system stimulation on leukocyte function. Now, I want to take a moment and really hammer home why understanding the interplay between our nervous and immune systems is so important. Stress is something that everyone encounters on a daily basis. By its nature, we cannot escape it. Large or sustained amounts of acute stress, however, is distinctly detrimental to our health. Therefore, understanding how stress of all kinds impacts our immune system can give us a greater understanding of how we become susceptible to disease. Now, if you also think that these are some important reasons to research this topic, go ahead and tap the like button. This brings us to the paper that we're focusing on today. This paper is called Adrenergic Regulation of the Vasculature Impairs Leukocyte Interstitial Migration and Suppresses Immune Responses by Devi et al. From the University of Melbourne in Melbourne, Australia. In this paper, the authors thoroughly examined the impact of the neurotransmitter noradrenaline on immune responses to provide a mechanistic insight into the relationship between the activation of adrenergic receptors and impaired immunity. At the beginning of their study, the authors hypothesized that sympathetic nervous system signals might modify the movement of T cells and tissues and lead to compromised immunity. To explore this, the authors injected fluorescently labeled T cells into mice and followed their movement. When looking at the normal T cell movement in the lymph nodes, they found that they moved in an amoeboid fashion. However, when given noradrenaline, the T cells abruptly stopped their movement and retreated their pseudopodia. This result was transient and immediate, as the T cells resumed their movement after 45 to 60 minutes. Now, noradrenaline is used to increase blood pressure for treatments of patients undergoing bodily shock, the treatment for which occurs over many days. So, to test the bodily relevance of this, the authors next investigated the impact of prolonged noradrenaline exposure. They found that a continual low dose of noradrenaline continued to cease T cell mobility so long as it was maintained. When looking at the effect of noradrenaline on the locomotion of T cells in non-lymphoid tissues, like in the liver and the skin, they also found this decreased T cell locomotion. Since noradrenaline signals through subtypes of alpha and beta adrenergic receptors, the authors wanted to identify which receptors cause a halt in motility. When the non-selective beta adrenergic receptor agonist isoprenaline was given to the mice in either single doses or multiple doses after cell movement recovery, there was a distinct halting of T cell motility. This activity was found to be specific because when this receptor was blocked, the T cells continued moving about unaffected. Surprisingly, the authors also found that isoprenaline not only affected T cells, but also halted the movement of B cells and dendritic cells in the lymph nodes. When looking closer at the beta and alpha receptors, the authors found that stimulation of beta 2 and alpha 1 receptors caused T cell locomotion to be halted. In particular, alpha 1 activity was specific because blocking it with an antagonist allowed for normal T cell movement. To see whether adrenergic receptor signaling on leukocytes or the surrounding tissue was responsible for inhibiting T cell motility, the authors isolated and then co-transferred normal wild-type CD8 T cells and those that lacked beta 2 adrenergic receptors into mice that were wild-type or also lacked beta 2 receptors all throughout their body. When the wild-type mice were given isoprenaline, their activity stopped. Remarkably, in the beta 2 deficient mice, administration of isoprenaline caused no effect. In their quest to fully understand how T cell motility is getting inhibited, the authors next looked at the vasculature. When mice were given noradrenaline, this caused a rapid constriction of the blood vessels in the T cell zone of the lymph nodes. When testing beta adrenergic receptor signaling, this effect was replicated. This also caused an acute reduction in blood flow rate after treatment. To examine the direct role of sympathetic nerves in this process, the authors created dread mice with a human receptor. This allowed for controlled stimulation of the sympathetic nerves with the small pharmacologically inert molecule C21. When wild-type mice were given C21, there was no effect. However, in the dread mice, this caused a rapid constriction of the microvasculature, correlating with an arrest in T cell motility. Because lymphocytes require oxygen to move about, and it is unclear how T cells respond to rapid oxygen changes in vivo, the authors next wanted to examine if blood flow affected lymph node oxygenation. They did this by using a probe that binds to cells under hypoxic conditions. They looked at CD8 T cells, B cells, and macrophages, and found when isoprenaline was administered, all of these cells became hypoxic, which was sustained consistently alongside T cell immobility. This effect was also seen with phenylephrine and with salmeterol, indicating a mechanism of action for why lymphocytes become immobile. Under hypoxic conditions, it was unknown whether leukocytes alter their calcium levels. To determine whether this was the case in the lymph nodes, the authors visualized calcium receptor activation. Administration of noradrenaline or isoprenaline caused a rapid, widespread wave of calcium signaling in leukocytes, which correlated with a loss of T cell motility. To directly assess the role of hypoxia, the authors superfused the lymph nodes with oxygenated media to circumvent it. Oxygen supplementation prevented the halting of T cell locomotion and the wave of calcium signaling following noradrenaline administration. The authors next hypothesized that loss of movement would be detrimental to the induction of protective immunity. To examine this, the authors used a well-established model of herpes simplex virus infection. Normally, the T cells cluster to the dendritic cells and become activated. However, with the addition of isoprenaline, T cell motility was halted for longer than uninfected mice, and the viral infection was sustained. In this system, when looking at T cells themselves subjected to sustained immobility, there was reduced proliferation and expansion of virus-specific CD8 T cells in the lymph nodes, and reduced virus-specific T cell recruitment to infection on day 6. In light of this, the authors also looked at the impact of adrenergic receptor signaling on viral clearance. They found that salmeterol stimulation caused a decreased cytotoxic activity against target cells. Now, because cytotoxic CD8 T cells are also important in cancer clearance, the authors also wanted to explore whether altered T cell mobility had an effect on anti-tumor responses. In short, it did. Salmeterol caused a decrease in cell proliferation, expansion, and activation. Finally, the authors investigated the impact of adrenergic stimulation on systemic immunity. They did this by infecting mice with Plasmodium burgii, a systemic parasite that causes CD8-mediated immune pathology known as cerebral malaria. When given either salmeterol or phenylephrine under these conditions, it caused an inhibition of T cell proliferation and expansion, a slight increase in parasitemia in the blood, and a significant delay in the onset of cerebral disease when compared with untreated mice. To quickly summarize and contextualize everything, during an acute stress response, the body releases a host of hormones, redirecting blood flow to the skeletal muscles and providing oxygen and nutrients for a fight-or-flight response. In this paper, the authors found that when the hormone noradrenaline or other beta-2 and alpha-1 adrenergic receptor agonists are administered, there was a constriction of the blood vessels. This constriction of blood vessels, particularly in the lymph node, caused a decrease in oxygen levels. In response to the hypoxic conditions, leukocytes sense this and undergo calcium flux, rendering them immobile. The lack of leukocyte locomotion corresponds to inhibited immune capacity, which impairs the ability of mice to fight off multiple types of infection and disease, ultimately indicating that stress causes impaired immune function. Not only do I think that these connections between bodily systems are exciting to investigate and learn about, they are also significant in a broader context. This information is significant because, quite frankly, we will always experience situations that activate our fight or flight response. Furthermore, periods of sustained stress are linked to onset of many different diseases and infections, like heart disease and reemergence of chronic infections. By understanding the impact of adrenergic receptor signaling on cellular functions, it may inform new developments for treatments of disease, and better inform how we currently treat health conditions. All science is basically a stepping stone for new knowledge, and these steps are driven by questions. And I had a few questions myself after reviewing this information. My first question has to do with the way that the experiments are conducted. It is great to see the effect of neurotransmitters, but how would the immune cell movement look under physiological periods of stress, or even different kinds of stress? I know it would be difficult to capture, but would leukocyte movement be inhibited in the same way, or are there different factors at play? Because we all experience different instances of stress in our lives, I would really like to see whether acute stress at different time points during an infection would have an effect on the outcome of said infection. That way, it could inform people on how to best conduct themselves during things like flu season. Additionally, because vasopressor treatments are a common treatment option for people with conditions that cause hypotension, I'm curious to know whether those people were more susceptible to common or uncommon infections. Thinking about it, Perhaps the data is already out there and just needs to be mined and compiled into a digestible unit. As always, though, my final question revolves around you. What sort of ideas or questions popped into your head when hearing about this information? I would love to hear about them in the comment section below. Also, let me know if there are any topics that you'd like to hear about in the future. Ultimately, I hope that you learned something, but more importantly, I hope you enjoyed your time doing so. If you did, give this video a like and subscribe for more in the future. Well, that's everything for today. Thank you for watching, and I'll see you next time.